Hello and welcome to the Biotech 2050 podcast. Biotech 2050 is a think tank chronicling the disruptions changing the biotech industry over the next several decades. Check out our website at biotech2050.com. I'm today's co-host, Alok Tayi. I'm the CEO and co-founder of Vibe Bio. Vibe is an AI platform that helps pharma companies make strategic decisions. I'm really excited today to be joined by Michelle Werner, the CEO of Alltrna. We're live here in San Francisco at JP Morgan 2026, meeting with a bunch of really exciting biotech and pharma companies and hearing about their strategies and view for the upcoming year. And so today we'll be having a really exciting dialogue about Alltrna, the work happening in the tRNA space more broadly, as well as what she thinks about the portfolio and the future for rare disease drug development. So, Michelle, thanks so much for joining us today.
Thanks for having me, Alok.
Maybe to kick it off, I'd love to learn a bit about your background and how you got to where you are today.
Sure. So I've been in pharmaceutical drug development for 20 plus years. I sort of happened into it right after university and then fell in love with the idea of bringing new innovations to patients. I spent most of my career in big pharma, BMS, AstraZeneca, Novartis, really focused on late-stage drug development and commercialization, and predominantly in the oncology space, which is certainly a therapeutic area that I call my first love and found very rewarding. As circumstances sort of changed for me personally, I have a child that was diagnosed with a rare disease that happened in 2020. And so as I was starting to think about, what do I do with this experience that I've developed over the course of my career, and is there a way for me to apply it in rare disease? That was really important to me because I could really see the unmet need, some innovation, but certainly a lot less compared to oncology, and really wanting to make a difference there. So that's where I sort of poked my head up and started to look at opportunities in that space, and I came across Alltrna. And Alltrna is a company that is really deeply focused in rare genetic diseases with our engineered tRNA technology. And for me, what's really special about tRNAs and rare diseases is that instead of this paradigm that we have even with novel technologies of single drug, single disease, which is really how it goes for mRNA, gene editing, gene therapies, all of the above, tRNAs are very special in that it could be single drug, many different diseases. So that to me really felt like it could have a profound impact on patient populations that are overlooked and underserved, and I really wanted to be part of it.
Yeah, it's amazing. Well, it also strikes me that there's this world where oncology is moving to a very precise and biologically defined tumor as opposed to geographic. And it sounds like maybe you might even be able to do something similar here with that sort of technology in the rare disease space.
That's exactly right. I do think there are a lot of parallels between oncology and rare diseases. Not always, but certainly especially, as you just said, genetic drivers of cancer have really transformed the way in a very targeted approach for treating those diseases. And I think we're doing exactly that, or trying to do exactly that, with many of the gene-based, RNA-based technologies. And yes, some of the development strategies may be similar, but I'd say, I think also even maybe the concept of polypharmacy and how we may ultimately be treating rare diseases in the future, I think that could be the sort of next phase of this.
Amazing. Well, certainly would love to drill into the biology of tRNAs in a second. Before we do that, one thing that's intrigued me about your background is the fact that you spent a fair bit of time looking at the commercial side of biotech and drug development. Yeah. It's rare to have someone with a commercial background work at an early stage company. What's one thing you think folks need to be thinking about that you need to be thinking about in an early stage biotech that you learned while doing commercial work in a bigger pharma company?
Yeah, so what I would say is that there's a lot of things that I learned in big pharma that are very much applicable to even an early stage preclinical biotech company. In a big pharma company, you do a lot of work to really develop a business case for an idea, to pitch that business case internally, secure funding for that from the executive team and the board. And that's exactly what we do from a fundraising perspective with investors in a small biotech. I also think that it's really important to really understand the market and the business opportunity and really trying to get a handle of that, which is actually not so straightforward necessarily in rare diseases. is definitely something that I think early stage companies should be paying attention to.
Yeah, it makes sense. You often meet a lot of folks who have deep scientific expertise in a space, but maybe overlook some of the other aspects of what it truly takes to get a medicine to patients. Now that said, pioneering a new modality takes a lot of gumption. Would love to hear a little bit about kind of the tRNA space as a whole, which I feel like has evolved since you even took the helm at Alltrna. What's exciting in that domain, and maybe just a little bit about the biology to help anchor the audience.
Yeah, so let's just talk about what a tRNA is, because many of us might not recall that from our Bio 101 days. Mm-hmm. So a tRNA plays a really important role in the protein translation process. So typically you have DNA that's transcribed into mRNA. mRNA translates into proteins. But what happens actually in the protein translation process, as the ribosome is sort of like going along the mRNA strand, inside the ribosome are these transfer RNAs. The role of a transfer RNA is actually to read the 3-letter code of each and every amino acid that makes up the proteins, to find that amino acid in the surrounding cellular environment, and then transfer it to the growing polypeptide chain. Hence the name transfer RNA, because it plays this important transfer function. But it really is the physical link between the mRNA and the protein, so it really speaks to the criticality of its role. Now, the way that we're thinking about tRNAs at Alltrna is actually to address a certain type of mutations that are called nonsense mutations. And I'm sure, Alok, you know that, but just as a reminder for the audience, a nonsense mutation occurs when you have, instead of one of these codes for an amino acid, it codes for a premature stop instead. And when you have a premature stop, the natural tRNA, there are no natural tRNAs that know how to read that. It reads it as a stop, protein translation stops too early, and you get a truncated or a shortened protein, and that's actually what causes disease. So the way that we're thinking about tRNAs, and actually now applying this preclinically, but soon to be clinically, is actually engineering tRNAs that know how to read these premature termination codon, understand which amino acid should have been coded for had that mutation not been there in the first place, and then transfer that corresponding amino acid to the growing protein chain, where the result then is a full-length functional wild-type version of the protein. So it's a precise correction of the mutation without making a permanent edit or change to the genome.
Yeah, amazing. And given the prospective potential of the platform and the modality, love to just understand a bit about how do you think about disease states, given that we have, again, a historical view of diseases being genetically defined, but not necessarily tRNA defined. Love to hear your thoughts.
Yeah, so as I mentioned with these tRNAs really addressing these premature termination codons, I think it's really important to recognize that the exact same premature termination codon occurs in many, many different diseases. In fact, we see it upwards of 4,000 or so different diseases. So unlike other technologies, which are for a single gene or for a single protein, because that's how they work, an engineered tRNA should be able to read through these premature termination codons that are the same across many different genes. What that effectively means is that a single one of our engineered tRNAs may be able to address hundreds, if not thousands, of different conditions. Now, when we're talking about starting with nonsense mutations to begin with, that actually occurs in about 10% of all genetic diagnoses. It stems from a premature termination codon. We would actually classify those patients as having stop-codon disease. So, they may have a different sort of genetic definition, but they actually have stop-codon disease because that's the underlying mutation that causes that genetic condition. Now, stop-codon disease represents about 35 million people worldwide, so quite substantial. And this actually kind of just really has an opportunity to change the way that we treat patients because, as you know, Alok, there are somewhere around 10,000 of these different rare genetic diseases. And right now, as we're tackling them one by one, that's a lot of diseases to get to. And I will share with you, certainly not just as the CEO of Alltrna, but as a rare disease parent myself, that patients don't have time to wait for 10,000 different innovations to become available for one person's perhaps condition. What an engineered tRNA may be able to provide the opportunity for is that patients theoretically across thousands of different life-threatening conditions may be able to benefit from the single engineered tRNA.
Yeah, that's amazing. Well, one thing I know we share in common is having rare disease children, and
Yes.
one of the pieces that struck me when I was involuntarily brought into this space was the fact that even though you might have a single label like Duchenne muscular dystrophy, you have many different genetic origins, right, that lead to a common manifestation. And so I could see thinking differently about the biology potentially transforming how we treat the class as opposed to just the individual flavor, perhaps.
That's exactly right.
Yeah, amazing. So now with that, tRNAs, exciting domain, obviously a new modality. I'm sure there's many folks out there who are early on in the development of siRNAs, ASOs, AAV gene therapies. As a pioneer in this novel modality and mechanism, any advice you have for the next wave of founders and CEOs trying to develop completely new modalities, especially given some of the risk aversion that exists in biotech?
Yeah, I mean, what I would say is, tRNAs really weren't an area of focus really before Alltrna came about. And now there does seem to be some interest in that area, which we can talk about if you'd like. But what I would say is, even when Alltrna was founded, we started with trying to understand the basic, like what's been done already in the oligonucleotide space, learning from siRNAs, learning from mRNAs. Of course, nobody has really done tRNAs before, but we didn't have to start from scratch everywhere. So there were some tools that we could apply, that we can leverage and apply in a different way than had been done before. There are other tools that we had to start over with that were unique for the tRNA. I'd say certainly when you're at a pioneer and doing something for the very first time, one of the best ways to try to tackle that is, try not to think about where you have to start from scratch everywhere, but maybe what you can do to really learn from lessons from before and focus on the things that are uniquely specific to whatever it is that's being pioneered.
Makes sense. Well, I was spending some time with some friends at Alnylam, and in addition to the work in siRNA, they were very proud of the work they did in delivery.
Yes.
And I'd imagine in the case of tRNAs, the novel modality delivery is probably part of the conversation. Love to hear how you're kind of thinking about that, and how do you tackle that, given that delivery broadly in drug development is still an unsolved problem?
Yeah, it absolutely is. So for our first development program that we will be advancing to the clinic this year, we call it AP003. That's an engineered tRNA. It's formulated in a lipid nanoparticle. We chose an LNP to begin with in the first instance because LNPs now have been in millions of people around the world. We know exactly how well they get to the human hepatocytes. We know very well what the safety profile looks like. So it actually takes some of the risk associated with the delivery off the table and really focus on the novelty of the tRNA as a payload. That was a strategic choice. Now, the great thing about a tRNA, however, is that it actually is pretty flexible and delivery agnostic. You can formulate it in an AAV, you can put it in other nanoparticles. We actually also believe that there's a way in which you can design tRNAs that are metabolically stable and can be conjugated, so leveraging maybe different peptide antibody conjugations that are also advancing on their own streams, and then maybe bringing these technologies together in the future. So there are lots of ways in which we could think about broadly applying a tRNA across many different diseases. Initially, if we were focusing on rare genetic liver because of the LNP formulation, but a tRNA would be, uniquely suited for CNS diseases or muscle diseases, and those are areas that we also are planning to explore as well.
Yeah, amazing. Well, with that said, you sort of alluded to briefly about the prospective clinical trial that you're hoping to kick off soon. Love to get a little more detail on Alltrna as a whole. What does the program, the portfolio look like today? And then would certainly love to hear about how you're thinking about the trial and the upcoming clinical work.
Sure. So I referenced AP003 already. That's what we call our lead development candidate. As I mentioned, it's an engineered tRNA formulated in LNP, and it's a tRNA that specifically targets the arginine to TGA stop mutation. Now, there are actually many different ways in which these stop mutations can occur. In fact, there are 19 possible amino acid stop combinations. There are actually two that are by far the most common. That's the arginine to TGA, as well as the glutamine to TAG. So our first program is the arginine to TGA. Our second program that's coming behind it is the glutamine to TAG. Those two alone represent 50% of all patients that have stop-codon disease. And then, there are less frequent ones behind it. So we're really focused on the two that we think can have the biggest impact, and that's where we really want to, where we're focusing on, certainly have demonstrated preclinical proof of concept for the arginine to TGA already. That's for the one that's going towards the clinic. We're doing that same work for the second one. And then we'll do clinical proof of concept for AP003 over the course of the next year.
Yeah, wow. And just on that note, one thing that struck me as interesting is that you mentioned there's only two circumstances where stop-codon disease primarily manifests.
Well, that are most common, 50%,
Most common.
yeah, exactly.
Okay, 50%. Got it. Biology rarely concentrates in that way. What do you think is unique about that particular pairing of amino acids that you think makes it the most
I mean, to be honest with you, I think it's really just a question of statistics, right? Okay. Just in terms of the different codes that make up different amino acids, there are 3 different stops. There's the TAA, a TAG, and a TGA. And then there are different amino acids. And if you look at the stop codon table, you can see that there are the likelihood of certain amino acids pairing with certain stops is the case, and that's why we have those 2 that are the most common.
Yeah, amazing, interesting. So that said, as you start to approach the clinic and you have this really unique way of defining disease, I'd argue to the first principles biology, right?
Yes.
How do you think then about clinical trial design, given that you're not using the same nomenclature or the same labels, right, that people often align to when it comes to both patients and regulatory authorities?
Yes, I mean, so we call it stop-codon disease because we think that's a great way to really characterize this very large patient population. We, of course, will also couple that with more familiar nomenclature before there's really I'd say a receptivity or an acceptance of stop-codon disease as an official term. But what I would say is actually, again, you talk about the underlying biology. When we are thinking about clinical trials, there are some novel ways in which we can develop these tRNAs that certainly has a playbook that has been used in other settings, which I think can be uniquely applied in the rare disease setting as well with the tRNA. And this is where I would describe basket trials, right? So if you kind of get to the sort of question that you asked before about the underlying biology and the sort of parallels with oncology, basket trials are actually used super commonly in the oncology space. Patients technically have different types of cancers. They're all identified by having their underlying causal mutation that is the same regardless of the cancer type, and then they're given one treatment in that clinical trial. We can actually do the same thing with a tRNA. So patients technically have different, I'll say, rare genetic liver diseases, and we're focusing on inborn errors of the metabolism in the first instance. And they could technically have any one number of inborn errors, but they would all be identified by having the same arginine to TGA stop mutation, and then could get AP003 in a single basket trial. That, is a playbook that is tried and true in oncology, has led to a number of different approvals there. We've actually seen it also in the rare disease setting, but because very few, if any, genetic medicines can be applied across multiple diseases, I don't think it has really been maximized, the potential there. Yeah. And I think a tRNA would be able to do that.
So that's super interesting. I'm curious then, as you start to approach the clinic, really practical or tactical question, like how do you think about recruitment, given that folks just aren't necessarily as familiar with the root biology of their indication as you all may be?
Yeah, so let's think about, so as I mentioned, we're going to be focusing on inborn errors of metabolism. Now, to identify these patients that have these nonsense mutations, actually those nonsense mutations would come up in the standard genetic sort of panel that would be done in order to be able to do the, either it's the whole genome sequencing or NGS or whatever it is, those mutations will get identified. Now, a patient or their family may not know that mutation because it hasn't been relevant before, but keep in mind that, inborn errors also are part of newborn screening panels very typically. Generally, the next step is to do the diagnostic workup, and the genetic testing would be a component of it. So there would be a question of looking into those genetic reports, making sure you can identify patients, and then those would be the patients that would be enrolled in the trials. Now, what I would also say is we're working very actively with the patient advocacy groups. They also have a mission to really make sure that patients have genetic testing done even generally. They think that's important information that might be important from a prognosis perspective or for other decisions. And so I think that there's a reason why these mutations can become more understood and well known, and obviously when there are treatment options that are available will become more important.
Yeah, wonderful. So that said, given the prospect for tRNAs, how are you thinking about the portfolio and the programs today? What does growth look like for you into 2026 and beyond?
Sure. So as I mentioned, we have 2 liver disease programs moving forward. One that we anticipate will be entering into the clinic this year. First time a tRNA has ever been in humans, so we're really excited about that. And then a second liver program for a different premature termination codon coming behind it. Beyond that, I actually think that it's going to be really important to demonstrate, what we're seeing in liver can actually translate to other tissues as well. And so we have some early work that's being done, I'd say really in the discovery phase in the CNS conditions as well as in muscular dystrophies, so muscle diseases, Duchenne muscular dystrophy in particular. If I just mention CNS, for example, there's a condition called Rett syndrome. It actually is a very devastating condition. 30% of all patients with Rett syndrome actually have an arginine to TGA stop mutation.
Wow,
so it's a really prevalent nonsense mutation of one kind. It's also a Goldilocks disease. So what that means is that if you have a technology that then overexpresses a protein, you may actually introduce a different but equally catastrophic disease.
Yeah.
You can't actually overexpress with a tRNA because of you're really just giving a tool back to the endogenous machinery to use. And so this may be an important strategy for this population.
Makes sense. And I know the existing standard of care folks are not super happy with in the case of Rett, so.
That's true, and I know that there are emerging gene therapies, and I guess we'll have to see what the data look like in those areas. But I think a tRNA could be a great approach. And then I mentioned also in the muscle space, especially Duchenne muscular dystrophy, and it's not just because I have a child with Duchenne, but think about these are massive genes and massive proteins. It's really the reason why we can only look at sort of micro versions of gene therapies, and an mRNA just is never going to be able to deliver that size of a protein. So a tRNA is actually quite small, right? And because it doesn't actually matter how big the gene is or how big the protein is, you just have to deliver a small tRNA.
Yep.
This may be one of the few, if only, approaches to restore full-length dystrophin, and that could be a game changer for this population.
Yeah, it's amazing. And do you think, again, coming back to our discussion around delivery, do you feel like, given the differences in the organ systems from CNS to muscular, I'm assuming maybe even difference in ROA, do you feel like there's more of an engineering challenge in kind of thinking about delivery that way, or do you find some commonalities there too? Yeah,
what I would say is the way that we're designing our tRNAs, there's actually 3 different ways that you can design a tRNA, one of which is really just a pure, simple engineering of the anticodon. I'd say that's level number 1. Then there's the sort of sequence optimization, so the 76 nucleotides that make up the tRNA, I'd say that's level number 2. And then there are chemical modifications, decorations that you put on the tRNA structures in order to be able to increase its activity and the potency and the stability. That's level number 3. We do actually all 3, and we think that different delivery mechanisms would be amenable to level 1 and level 2, let's say from an AAV perspective. But actually a conjugation, you can do all 3 in an encapsulated technology like an LNP. You can do all 3. And I think that creates some different design elements. But what we've learned is that in a true platform approach, a lot of the learnings and how you design one can be applied to how you design another. And so there's a lot of efficiencies that are realized.
That makes sense. And I think formulation and delivery engineering risk is a different bucket that's perhaps a little more tractable than, say, pure biology, et cetera. So that said, would love to ask you a couple quick rapid fire questions about the topics du jour. First, AI.
Yes. Well, we use AI in our tRNA design process, and that's important actually because the way that tRNAs are designed, there are actually more tRNAs than there are atoms in the universe, so a huge combinatorial space. So we really use AI in order to be able to help sort of in a generative way predict which ones are going to be most effective. So that's what's the bread and butter today. I'm actually really curious that as we transition to the clinic, how AI can help us there, and I think certainly from a patient identification perspective, that will be playing a role, and I'm looking forward to learning more from others who are in that space already.
Yeah, indeed. Next question, China.
China, big topic. Yeah, so I actually think that there's work, like I want to understand how we can leverage China in an appropriate way. I mean, I don't think we should be thinking about do you or do you not. I think it's really just a question of how. So I think that if there are ways to accelerate drug development and create some efficiencies in the process, I'm all for that.
Yeah. All right, and the last question, which I know you probably have strong opinions and passion about, is rare disease. What does the future look like?
Wow. I mean, I definitely think we're heading towards a direction of like ultra personalized approaches. We've seen a lot of really interesting stories over the course of the last year of these successful N of one, like baby KJ-like stories. I think for me now it's all about how do we take the baby KJ and scale it in a way that hasn't been able to be done with rare diseases. I hope that's where an engineered tRNA plays a role, but that's what I'm excited about.
Yeah, well, I think that also comes to the more recent kind of developments at the FDA around new mechanisms, new pathways, like plausible mechanism. Love to kind of hear your just overview or take on that, and how you think the process of developing medicines is going to evolve and the operating model for biotech changes because of it.
Sure. I mean, I definitely think there's an opportunity, and this is what plausible mechanism is getting at, is like if you really understand the sort of causal biology behind a disease, and if you have a good understanding of the sort of mechanism that could address that causal biology, this is where plausible mechanism really comes into play. So I do think there's a strategy whereby we can get much more efficient in terms of what we need to demonstrate preclinically from a safety and efficacy perspective, maybe perhaps actually eliminating the need to have very robust animal models, and hopefully being able to get into patients in a much faster way. I think we need to wait to see what that guidance ultimately looks like, but I'm really excited about the possibilities.
Well, indeed, and given the quantity of rare diseases that are out there, obviously we need all the help we can get.
Yeah.
If you were to think about starting a company with such prospective guidance in place, how do you think you would operate differently than you might say even in today's world?
What I would say is, I think what we're learning is that having really early dialogue with regulators is becoming more and more important. Especially as some of these guidances are new and they're evolving. I think that there is a narrative and a dialogue and an education that needs to happen at very early stages in order to be able to bring all of the stakeholders that we need to get drugs to patients at a much earlier stage. So I'd say don't, glaze over that. Try to make that part of your strategy at a very early start.
That makes sense. Well, I know that was something that you all did very early on,
Yeah,
right?
did.
Is to engage and be prescient about the importance of that. So wonderful. Well, I think it's safe to say that we're all eyes on the new program once it goes into the clinic and the IND, and very much wish you all the best, and certainly love to hear kind of the updates and the live experiences as they come through.
Yeah, we'll tell you about it later this year. Thanks so much, Alok.
Thank you so much.